Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Syahirah binti Mohd Khalid with the metric number of 2012152. For today's video, I will be explaining the topic of metagenomic sequencing. So before we dive in deeper into the topic, let's try to understand what is the meaning of metagenomics. So metagenomic is the study of genetic material of entire communities of microorganisms recovered directly from environmental samples. Obtaining bacteria in pure culture is typically the first step of investigating bacterial processes. However, standard culturing techniques account for 1% or less of the bacterial diversity in most environmental samples. The principles involved in metagenomic is DNA amplification. DNA amplification occurs when a short segment of DNA is amplified using primer-mediated enzymes. So let's move on to the metagenomics workflow, where the samples are collected from a direct environment, such as soil. Next, the DNA is extracted from the sample through chemical lysis. Then, extracted DNA is fragmented into smaller pieces, and adapters are ligated to the ends of these fragments to facilitate sequencing. DNA fragments are then amplified using PCR. High-throughput sequencing technologies, such as Illumina, are used to read the DNA fragments. Next is bioinformatics, where quality control is done to filter out low-quality reads and adapter sequences. Lastly, the taxonomic and functional profiling of the sequence DNA is analyzed. So there are two techniques of metagenomic sequencing, which are shotgun metagenomics and also targeted metagenomics. So shotgun metagenomics involve randomly breaking up all the DNA in a sample, while targeted metagenomic focuses on sequencing specific regions of the genome. Next is the advantages of metagenomic sequencing, which involve culture independent, providing comprehensive information, and also can detect novel and rare pathogens. The challenges of metagenomic sequencing is complexity and also assembly difficulties. Lastly is the metagenomic sequencing is applied in various fields such as environmental monitoring, human health, microbial ecology, agriculture, and also biotechnology. Application of metagenomic sequencing in human health helps in investigating the human microbiome's role in health and disease, and to detect drug resistance genes for pathogen. So that's all from me. Thank you.